Welcome to today's video webcast event, Gain Therapeutics KOL Analyst Event, Beyond Symptomatic Treatment of Parkinson's Disease. Today, you may submit online questions at any time using the window on the webcast. Please note that today's event is being recorded. Before we begin, I would like to read a brief statement. Some of the statements during the course of today's event will be forward-looking statements. These forward-looking statements are based on the company's expectations and assumptions as of the date of this event. Each of these forward-looking statements involves risks and uncertainties that could cause the company's future results to differ materially from those expressed or implied by these forward-looking statements. So the company is providing here the customary safe harbor disclaimer regarding these forward-looking statements. At this time, I would like to turn today's conference over to Hartaj Singh of Oppenheimer & Company. Please go ahead, sir.
Great. Thank you, Chris. Thanks for making this easy. These things are never easy, actually. So thanks for making this as painless as possible. I'm one of the biotech analysts at Oppenheimer & Company. My name is Hartaj Singh, and our team covers Gain Therapeutics. Today, we are actually very lucky to have three great speakers ahead of us. We have presentations from Professor Roy Alcalay, Professor Tiago Fleming-Outero, and Dr. Joanne Taylor, who's with Gain Therapeutics. And they will go in that order. Our first speaker is Dr. Roy Alcalay. He's the Chief of Movement Disorders Division at Tel Aviv Medical Center in Israel. He holds an associate professorship in clinical neurology at Columbia University. His research focuses on biomarkers and genetics in Parkinson's disease, and he's a member of the Michael J. Fox Foundation-funded Parkinson's Progression Markers Initiative Executive Steering Committee and the PI of the Parkinson's Foundation-funded PD Gene Ration Study. Dr. Roy Alcalay, thank you for taking the time to join us, and please take it away.
Thank you, and thank you for inviting me to speak today. So I was tasked with a relatively simple task of a talk, and that is to talk about GBA-associated Parkinson's disease and other synucleinopathies, current unmet medical needs and future therapies, or in other words, why focus on GBA Parkinson's disease? So when these are my disclosures. I'll give you a second to take a look at them. And I will start by saying that looking at genetics as a source for finding a new treatment for a disease sounds very promising and has proved itself in other diseases. There's now in ALS, SOD1 mutations are associated with a precision medicine drug. In SMA, there is a drug that is working, which is gene specific. So looking at the genes to look at new novel targets sounds very trivial, very intuitive. Now, why look at GBA? There's 90 hits on the GWAS, on the Genome Wide Association Study for Parkinson's. This is taken from Naz and colleagues from 2019. So when we look at the genes for Parkinson's in this figure, you'll understand why GBA and LRRK2 pop up. In the x-axis, we're looking at how frequent are the mutations in the general population. And in the y-axis is if you have the mutation, what's your risk for Parkinson's? And the vast majority of genetic risk factors for Parkinson's aggregate in two places. Those in the bottom right, they're colored green here, are variants that are relatively common, so 20-30% of the population can have them, and they increase the risk by 20% or 40%. So instead of 1% of people with PD, 1.2% of those who are carriers. So very common, but the effect size of a mutation or a variant is not that high. At the top left, there are variants or mutations that if you have them, your risks for Parkinson's is very, very high. Like people who are homozygous carriers for Parkin, almost all of them will develop Parkinson's. But these mutations are exceedingly rare. LRRK2 and GBA are located somewhere in the middle, where they're relatively common enough to start thinking about clinical trials, and they're risk enough. They have enough of an effect size that it makes sense to modify this effect to reduce Parkinson's risk or to intervene on it. And that is why I think GBA as a gene is a great drug target. So my brief talk will be focused on why focus on GBA. And I'm basically going to talk about 5 points. One first is that pathogenic variants in GBA are the most common genetic alterations identified in Parkinson's disease. The second is that GBA or GBA1 in its new name, is very similar to idiopathic Parkinson's disease. So clinically it looks the same, which holds promise that treatments for GBA may later be helpful for people without GBA. And when we look at them under the microscope, that's the third point, the diseases look very much alike, the genetic and non-genetic. The fourth point that makes GBA an interesting target is that we know what happens when people have 2 mutations in GBA in the disease that is called Gaucher. And a lot of research from the Gaucher world is research we can use now for understanding GBA PD. And that there have been already clinical trials for GBA PD. So they've demonstrated feasibility already. So it's not like, oh, this is not doable. Not only that it's doable, people have done it already. So starting with the fact that there are the most common genetic alterations in Parkinson's, I will show some information from the PD Generation study. The PD Generation study is funded by the Parkinson's Foundation, and its goal is to make genetic testing and counseling accessible for people with PD and for their doctors. The idea is that if people, that this is genetic data of people, if they don't know it, they won't know how to act on it. And specifically for the talk today, if people don't know that they're carriers, they won't participate in the study. And we realized that once we offer people genetic testing and counseling to explain to them the genetic results, a lot of people are interested in getting and obtaining the genetic results, and they basically line up to participate in the study. So the cut data of October 2023, after less than 4 years of study, roughly 10,000 people have been genotyped and received the results back. Out of those nearly 10,000, 10% or so, 12.6, received a positive result. The vast majority of those who received a positive result received it in the GBA1 gene. So 7.2% of all comers, and this is unbiased recruitment. We've encouraged anyone to participate. 7.2% of people with Parkinson's disease harbor a GBA mutation that would make them eligible for a GBA1 intervention. In this case, all of them received the results back, and I assume that some of them are just waiting in line for a clinical trial to start. So the second point, so now that I showed you that it's relatively common, we have at least from one study from PDGene, 700 people who know their genotype, and they were interested in knowing their genotype so they may act on it, acting by means of participating in studies. Let me tell you a little bit about the phenotype of people with GBA1 Parkinson's. So people with GBA, so just a word back, as I said, roughly 5 to 10% of people with Parkinson's carry a GBA1 mutation. In selected populations, specifically Ashkenazi Jews, the risk is much higher. So over 20% of people of Ashkenazi Jewish ancestry with Parkinson's carry a GBA mutation. But carrying a GBA mutation doesn't mean Parkinson's. It means that it increases the risk for Parkinson's. So the penetrance is estimated at roughly 10 to 30%, depends on the studies. Probably closer to 10 if you ask me. Once people develop Parkinson's, their Parkinson's progresses in a faster rate. So this is a paper from Celia and colleagues. It's an Italian study that looked at outcomes that are considered bad outcomes, dementia, death. Other people showed it with motor outcome. And we see that people with GBA mutations progress faster, which means they reach those bad outcomes of dementia or death faster than people with Parkinson's without these mutations. And they also showed a dose effect, that when we'll talk about it a little bit later, when they have a severe GBA mutation, and one that is more, that is probably bad for you, worse for you, the time to reaching dementia or death, and others showed it with motor performance, happens faster the more GBA burden you have. So idiopathic PD progresses the slower, mild mutation faster, severe mutations even faster than that, and 2 mutations the fastest. When we look at it as a clinician, I'm also a clinician, on an individual level, I cannot take a look at someone and say, oh, you have a GBA mutation. But if we look at people as a group, they have Parkinson's that looks like Parkinson's progresses a little bit faster. Parkinson's nowadays is not just defined by the motor symptoms that people think about when they think about Parkinson's, like tremor and slowness. There's actually non-motor symptoms, a battery of non-motor symptoms associated with Parkinson's, including cognitive impairment, hyposmia, reduced sense of smell, acting out one dreams, which is REM sleep behavior disorder, and autonomic dysfunction that would be clinically presented with constipation, lightheadedness when changing positions. All these symptoms are common in idiopathic PD, but actually even more common in people with GBA mutations and Parkinson's. When we want to look at the ultimate phenotype, not just how did the person experience the Parkinson's, but also and how did the clinician perceive their Parkinson's, but when you look under the microscope, what do you see? We see that the Parkinson's in GBA PD is very similar to idiopathic Parkinson's. So I'm showing you two slices from the brain. One of them on the right, which is the pink, is stained in the typical staining of the brain, which is called hematoxylin eosine. And in this one, if you see it just in the middle, you see a Lewy body. The Lewy body is accumulation of protein, specifically alpha-synuclein, in the cells. That's typical for idiopathic Parkinson's disease, but this brain is taken from someone with GBA and Parkinson's. And when we stain with antibodies specifically against alpha-synuclein, and that's the staining to the left, we see that these Lewy bodies in a person with GBA PD are indeed positive for alpha-synuclein. In two studies, one from England and one from Columbia University in New York, when the autopsies of people with GBA PD were compared to idiopathic Parkinson's without the GBA PD, those with GBA mutations had more burden of this protein of alpha-synuclein and more Lewy bodies in their brains. So I hope I convinced you that the GBA is relatively common among idiopathic PD patients. Clinically looks the same, progresses faster. Progresses faster is bad news for the patients. Good news for people who design clinical trials because outcomes can be reached faster. But it's not just clinically, also under the microscope, the diseases look alike. The next point that I wanted to make is that because GBA is not a weird gene that nobody heard about it before. GBA was well known because if you inherit two mutations in GBA, people develop the disease Gaucher. It's a common lysosomal storage disease. In addition to Fabry, Gaucher and Fabry are the two most common lysosomal storage diseases. The lysosome, I'm sure that we'll talk about it a little bit later, is the recycling bin of the cells. That's where proteins that need to be taken down or lipids that need to be broken down and reused are shipped into and rebuilt. In Gaucher disease, the enzyme, the GBA enzyme, the glucocerebrosidase enzyme that is called GCase, isn't functioning because there's two mutations in GBA, and as a result, lipids accumulate. Gaucher is divided to two types, three types. Type 1, which is more common in Ashkenazi Jews. People can live till an old age, and the reason is because those lipids accumulate in the areas in the periphery, not in the brain. The lipids accumulate in the liver, the spleen, the bone marrow, and we know exactly which lipids accumulate, the lipids that GBA, the glucocerebrosidase, needs to break down. If the disease starts or is present in the brain in ways that is not Parkinson's, it's called neuronopathic, and then type 2 and type 3. Type 2 is in infants, type 3 is in older kids. A disease develops that is very similar to type 1 with the involvement that I mentioned, in addition to brain involvement that can cause severe neurological presentation with seizures, myoclonus, cognitive impairment, and ataxia. When we look under the microscope for people with Gaucher disease, we see this is a macrophage. We see the macrophage in the center that accumulates the lipids. So we understand a lot of what happens when we don't have the GBA enzyme, the glucocerebrosidase enzyme, work. When the glucocerebrosidase enzyme is completely deficient, lipids accumulate. We know exactly which lipids accumulate, glucosylceramide and glucosylsphingosine. And the good news about Gaucher is that Gaucher has treatment. The problem, the reason why we can't try to use Gaucher drugs for Parkinson's is that retreatments for Gaucher is given peripherally to the vein or orally, but it doesn't penetrate the blood-brain barrier, so it wouldn't reduce the burden of the reduced glucocerebrosidase in the brain. So just a quick look at the metabolic pathway of GBA. This is the ceramide metabolic pathway. It happens in the lysosome, and when glucocerebrosidase, circled in purple, isn't working, the lipids that it's supposed to break down accumulate. Glucosylceramide and upstream pathway lipids, and also glucosylsphingosine that is not in the picture. In red, you see other lysosomal storage diseases, Krabbe's disease, Niemann-Pick, and Fabry. All of them were associated with the increased risk for Parkinson's, but none of them is as common and as big of a burden as Gaucher disease. So the thought is, like, what links the GBA mutations and to Parkinson's? And the most intuitive hypothesis to propose is that it's because of a loss of function. Loss of function means you have a mutation in the GBA gene, the protein doesn't work as well, the enzyme doesn't work, and that's why Parkinson's risk is increased. The reason to support this loss of function hypothesis is that there's over 200 mutations in GBA, and many of them have been associated with Parkinson's. So it's not that it's one mutated protein or another, it's all of them. Like, once there's a problem in the GBA gene, the risk for Parkinson's goes up. And another reason to think that it's loss of function is that if the mutation is severe, so there is less activity of the enzyme, then the risk for Parkinson's is higher. And as I showed you, the severity of Parkinson's when you have the mutation is even higher. And also, mutations that don't produce anything, that are called null mutations, are also associated with Parkinson's. So arguing again that less of the glucocerebrosidase enzyme, the higher the risk. I just want to say that this theory is not pure. There's other reasons to think that there's more than just loss of function of the glucocerebrosidase enzyme. And these are the gain of function hypothesis. The reasons to think about that is that I told you that Gaucher patients are at increased risk for Parkinson's, and maybe start even in a younger age than people without Gaucher. So people with Gaucher, they have 2 mutations in GBA, and still the majority of them will reach old age without developing Parkinson's, even though they have very little enzyme activity. So it may be not just the enzymatic activity, there is something else going on. And then when we look at the microscope, under the microscope at the brain, as I showed you in the previous slide, we see that the changes in the brain of people with GBA and Parkinson's look like Parkinson's. There's Lewy body with accumulations of protein. We don't see the accumulation of lipids that one would have expected if the problem is that the enzyme is not working as well. So a few more words about the glucocerebrosidase. It's an enzyme that is reduced, that is completely diminished in Gaucher. People have shown in multiple studies from competing groups in different continents that reduced GCase activity, glucocerebrosidase activity, the enzyme that GBA encodes for, is present in autopsies of people with idiopathic PD, even with GBA mutations. That would make sense, but also people with idiopathic PD without GBA mutations. In the blood, when we try to measure it, the results are not as conclusive, but there is mild reduction, at least in a couple of studies, in glucocerebrosidase activity in idiopathic PD compared to controls. In people with GBA mutations, there's clearly a reduction in glucocerebrosidase activity. And there is some hints in the literature that the reduced activity is also associated with more severe motor phenotype. So the next point that I wanted to talk about, why focus on GBA1 and what's missing, is that precision medicine studies in GBA1 have demonstrated feasibility. And I'm sorry, in this slide, it was the animation is a little bit broken, but the background slide is from a review that I wrote with Suzanne Schneider to talk about the different potential therapies for GBA. And I think the take-home message from my talk, I hope that will convince you, is that GBA is a great target. How to target it is open for questions. So one hypothesis, I'm going from the left to the right, one option to how to treat the GBA pathway is, instead of making the GBA enzyme work, the glucocerebrosidase enzyme work more, let's make the counter enzyme work less. And that's the glucosylceramide synthase. And there was a big phase 2 clinical trial led by Sanofi that showed feasibility. They recruited hundreds of people with GBA mutations around the world, Europe, Israel, Japan, North America. And the study didn't show that the drug worked, but it showed that we can do precision medicine in GBA PD. It was just published in 2023 in Lancet Neurology. And other ways to target the pathway would be to increase the enzymatic activity. And that's the second box, and the first box in green. That works well in Gaucher. The problem with that for Parkinson's is that those treatments currently don't penetrate the blood-brain barrier. The enzyme replacement therapy that is used for Gaucher is too big to cross the blood-brain barrier, and therefore currently that's not an effective way. There are the second box to the right would be gene therapy, like inject a healthy gene with a gene with a healthy GBA gene to the brain. There is an active recruitment for a study of the compound mentioned above. And the last the mechanism that I would like to mention is small molecules. These small molecules can be called chaperones, not all of them are chaperones, that can either take the GBA, the enzyme, from the endoplasmic reticulum to the lysosome where it belongs in the recycling bin of the cell and make it work, or just for one way or another make the enzyme work faster or better. There's one drug that was developed by LTI that is now in clinical trial in North America and Europe, working exactly in this mechanism. And the Gain Therapeutics compound that will be mentioned later is also targeting this path here. The idea is that currently people with Parkinson's disease are diagnosed clinically and getting treatment by symptoms. I hope that this is dated and will change soon enough to a very different paradigm, where people with Parkinson's disease will get a clinical diagnosis, will get a biomarker study, like a study of alpha-synuclein that is now works very, very well from spinal fluids, ideally from peripheral tissue in the near future, and brain imaging. Based on this, they will receive a Parkinson's diagnosis. Based on the Parkinson's diagnosis, they will get genetic testing. And based on the genetic testing, they will get the tailored treatment. And the first group that I would target is the GBA1 carriers, is because that's the largest genetic group. If a drug works in the genetic group, the next phase, and that will be the last box to the left, will be to look at non-carriers of the gene and look at their biochemical portfolio, like their biomarker signature, to see whether they have biomarkers to suggest that one of the genetic drugs would work for them. So to summarize, I think that the future holds a lot of promise in precision medicine and Parkinson's. We've gone a long way. One of the things we've done is to make the genetic data accessible for people with Parkinson's so they can choose to participate in studies. Pathogenic variants in GBA are the most common genetic alterations identified in Parkinson's disease. There's a couple of international studies that help people get the results back. There's no FDA-approved intervention on the GBA pathway. And actually, there's no FDA intervention approved in Parkinson's to slow down disease progression. The treatments that are approved by the FDA are symptomatic. Precision medicine drugs in other neurodegenerative diseases that I mentioned, like spinal muscular atrophy and ALS, have been approved by the FDA, so it's not inconceivable. It's just a matter of time till it reaches PD. And from all the genetic targets, I think GBA and LRRK2 are the most promising because of the prevalence of mutations. So thank you for listening.